It's a pleasure to be on this webinar and to give an update on the latest developments at Thermo Fisher in the field of cryotomography. So, say after the interesting previous talk looking into liquid electron microscopy, where you can look at dynamics, I now move back to the field of cryo-electron microscopy to show you, let's say, where we are at the moment in cryomicroscopy, and then especially for cryotomography. Over the last decade, there has been a big revolution in the field of cryo-electron microscopy, and especially in the field of single particle analysis, to look at the structure of isolated proteins, viruses, and macromolecular machines. And this has been mainly due, say, to the development of more stable microscopes, but also automated microscopes, and especially over the last couple of years in the development of new and better detectors. Now, if you look at the workflow of cryo-electron microscopy, then of course it always starts with the sample preparation. And for single particle, that means that you need to isolate your proteins or macromolecular machines out of their cellular context, get them in solution, and then in order to visualize them at high resolution, we need to vitrify the cells. So we have a vitrification robot where you make a very thin film of the isolated proteins in the order of 50 to 100 nanometers, depending a little bit also on the dimensions of the complexes you want to look at. Then you collect the data fully automatically. So nowadays on these microscopes, which are equipped with an autoloader, you can load 12 samples at a time and then image fully automatically all those 12 samples during the night or over the weekend. Due to all the automation, and especially also due to the development in detectors, data acquisition is now much faster than a few years back. On a standard sample like apoferritin, which was also mentioned in the previous talk, let's say within an hour data collection, you can already get resolutions below 2 angstrom. So that's really revolutionary compared to 5 to 10 years back. But then when you have collected all your data, all your single particles, you still have to do quite some work to get a 3D model using different kind of reconstruction softwares. Now, if you look at the timeline, then typically within 1 to 2 days, you can get high resolution structures of many different samples. And just to give you a few examples of what is currently possible in the field of single particle electron microscopy. The top row are data from the Titan trials with an energy filter and a Falcon 4 detector. And here, for instance, a very popular receptor for drug development, the GABA receptor, isolated and then resolved to 1.7 angstrom or 1.9, where you can really see where a drug, and in this case it's zopiclone, where that binds in the binding pocket of this GABA receptor. Nowadays we can also go to very small proteins. This is human hemoglobin. Here an example of a SARS-CoV-2 membrane protein. So during the pandemic, cryo-EM was used quite a lot to resolve the structure of the membrane proteins of the ion channels in order to develop these vaccines. And in the bottom row, a few examples which were taken on the glacials, which is a 200 kV microscope with the same outer lower and also with the same energy filter and detector. And you can see that also on this system, for instance, for the GABA receptor, you can easily resolve that below 3 angstrom, so to 2.4 angstrom resolution. So this was all on single particle. That has yielded, let's say, a lot of results of isolated proteins, structure determination for drug development. But still, you have to isolate the protein and take it out of its natural environment, take it out of the cell. So therefore, it's very interesting to look at these proteins in their cellular context. And that's where cryo-electron tomography comes in view, because there you can look at these proteins in their natural environment. So we believe, let's say, that the next technology push and the next revolution in cryo-electron microscopy will be in the field of cryotomography. And there are also a few indicators for that. If you look at the high impact journals, like Nature Methods, where it was mentioned as the method of the year in 2020, but also in other journals like Science and Cell and PNAS, there's more and more interest in cryotomography. And also, if you look at the number of publications, you can see a steep increase in the number of publications on cryo-electron tomography. And if you compare that to the number of publications and EMDB data sets, it's comparable to what happened to single particle, let's say 5 years back. So we see a steep increase in the number of publications using cryo-electron tomography. But cryotomography can be done on different types of samples. For instance, if you look at viruses and bacteria, then most of the time these are, let's say, thin enough to image them directly into the electron microscope. Viruses range from 20 nanometers to 150 nanometers. And that's still thin enough to image in an electron microscope, because that's one of the disadvantages of electron microscopy, that you need a thin sample. But if we are interested in where proteins are located in cells or even in tissues, then these samples are far too thick to image directly into the electron microscope. So for these kind of samples, let's say after vitrifying these cells, either by plunge freezing or by high pressure freezing, we have to thin those cells. Now, in the past, people tried to do that by cryo sectioning, making vitreous sections. That appeared to be extremely difficult. So nowadays, that is mostly done by a focused ion beam to thin those samples. So how does that workflow then look? You can culture cells on a grid, and for this whole workflow, basically we start immediately on the outer grids that also go into the cryos or the glacials. Then after the vitrification, in most cases, you want to know where you want to mill to get your area of interest into such a thin lamella. And this can be done by correlative microscopy. So for instance, if your protein is fluorescently labeled, or if you add a fluorescent label to your cells to identify, for instance, mitochondria or the nucleus. Then after the freezing, you can take a fluorescent image under cryo conditions to pinpoint to the area where you want to mill later on. So then you transfer your sample from the cryo-light microscope into the dual beam system, all under cryo conditions. And then you can use the ion beam to thin down and to mill your cells. And then after the milling, you transfer the cells to the electron microscope, and you can record your tilt series and reconstruct your tomogram, or do subtomogram averaging to get high resolution structures of the individual proteins in a cell. So that was schematically. How does that look in real life? So here on the left, you see our Aqueous dual beam system, which has an SEM column for imaging and a gallium FIB to thin down your cells. It has a cryo stage that is fully rotational, and there we can thin down the cells. So that looks like this. You define a window, and then with the gallium beam, you ablate material from your cell until you're left with a thin lamella, which is still held by the remainder of the cell. And typically you aim at a thickness of 150 to 200 nanometers. Then that sample is transferred to the electron microscope, and there you can collect your tilt series. So in order to do the 3D reconstruction, you need to tilt your sample over a wide tilt range to get, let's say, all the different projection images from the sample that you have. And then by means of reconstruction software, you can reconstruct the volume. And here you see an example where the interior of a cell was reconstructed. So you can nicely see the Golgi areas, vesicles, budding from the Golgi, and also you can see ribosomes. And if you look more into detail, you can see marked tubules and actin filaments. So this is our Aqueous 2 system for sample preparation for cryo-electron tomography. This system has a few advantages and unique things. And I will go over them in the next couple of slides. So this is how the system looks schematically. So here we have a rotational cryo stage, which is cooled down to minus 170 degrees. Here you see the focused ion beam, so the gallium column, the SEM column. We also now have an integrated light microscope. Here we have the sample on a loading station. Then we retract the sample from the loading station. We put it on the load lock here. Still, your sample is cold. We vent it, and then you can insert your sample into in the chamber of the dual beam system. And then with the gallium, you can thin down your sample. And in the meantime, you can do imaging with the SEM column. Now we've put a lot of automation into the system because, especially also for subtomogram averaging, you want to prepare as much lamellas as possible to collect as much tilt series as possible. And this is now fully automated. So in the software, you can define the regions and define the milling boxes where you want to mill the lamellas. So here you see an example on a grid where we have milled lamellas on several positions in the grid. So that helps you to get a lot of lamella in an overnight run. Now, we can also do cryo slice and view, where, say, with the gallium beam, you remove a thin layer from your frozen material and then take an SEM image, remove another layer, take an image. In that way, you can also build up a three-dimensional image in space, but it can also help you to guide you to the area of interest where you want to take your lamella. So it provides, let's say, the contextual image. And then, for instance, if you're interested in the nucleus with the slicer view, you slice away material until you are in the nucleus, and then you can start preparing your lamella. We also have a lift-out system, so especially for bulk samples, and bulk samples can mean a cell culture, where you look at the cell, or even at a small piece of tissue, which has been then vitrified by high pressure freezing. You can mill out a trench, and then with a cold needle, you can bring that to another half moon grid and then thin it down further. So in this way, you can remove small pieces from your sample, from a bulk specimen, and then thin it down to be able to do the tomography workflow. Now, the very nice thing of this Aqueous 2 is that it can be equipped with an IFLM, which is an integrated fluorescent light microscope into the chamber. Because as you saw previously in the workflow, there's still many transfer steps from your sample. So after vitrification, you have to transfer your sample to the Cryo-Correlative Microscope, then transfer it to the dual beam, and then after thinning and milling, you have to transfer it to the electron microscope. And of course, each transfer step has a risk of contamination or damaging your sample. So that was the main reason to integrate the fluorescent light microscope into the chamber of the dual beam, where we now can do the fluorescent imaging and the SEM imaging on the same sample. So here you see a schematic. We have an LED light source, a CMOS camera. This is all outside of the microscope. Then there's a vacuum window, and then let's say in the vacuum, close to the sample stage, we have the objective lens to do the fluorescent imaging. So here on the left, you see an image taken with the ion beam, and here on the right, top right, you see a combined image of the ion beam image with the fluorescent image. And that will then help you to target the area of interest to prepare the lamella. So here you see the system in real. Here you see the SEM column. This is the gallium column for the fibbing. This is the needle for the lift out, so the easy lift. And here you see the light microscopy module. So here is the LED source and the CMOS camera. So now a few examples to let's say to identify your area of interest for the milling. So this is an SEM image of cells grown on a grid. Here you see the quantifoil. And then we can overlay that with a fluorescent image. Since this is all integrated into the correlation software, and we know exactly where on the stage position we took the image, we can easily overlay the two images. So the SEM image with the fluorescent image, and this is a projection stack of the fluorescent imaging with the SEM image. So then you can identify your area of interest. In this case, the mitochondria were labeled with mitotracker green. So here we have an area with quite a few mitochondria. Then you can prepare your lamella in this region. And then even after preparing the lamella, you can go back to the fluorescent imaging and check if your structures of interest are still present in the lamella before you thin it down to the final thickness. So this is a fluorescent image, and you can see that we're still in an area where we have quite a few of mitochondria. It can also help you to identify the area where you want to take your tomograms later on in the cryo-electron microscopy. Here's another example. So here you have the SEM image. Here you see the cells grown on the grid. An ion image, so an image taken with the gallium beam. This is already in tilted view. Then we can project the fluorescent image on top of it, identify the area of interest for the fibbing, and then start drawing the boxes in the software to identify exactly where we want to mill that thin lamella of roughly 200 nanometers. So here you see a real example of that. We identified the cell of interest and the area of interest. You start milling the lamella. So first we do a coarse milling, that we have a lamella, in this case of roughly 10 micrometers, or 5 micrometers. We can take a fluorescent image again to see if our area of interest or molecules of interest are still there. And then finally thin it down to the final lamella thickness of about 200 to 250 nanometers. And this is then how these lamella finally look in the SEM image. And here you see a corresponding TEM image. And again, you can overlay those images with the fluorescent images. And now you can identify the areas where you want to take your tomograms. So I hope I've shown you that, let's say, with the Aqueous 2, with the dual beam system, we are able to prepare samples for cryo-electron tomography, either by on-the-grid lamella milling, or by using the lift-out method and then thinning these small pieces of tissue or so to the final thickness. This is all due to all the integrated software in the system, so that allows us to do automation. You can do overnight runs, but it also allows us to correlate the fluorescent images directly with the SEM images and the FIB images for the targeting. This is a final example of how such a tomogram looks. This is from chlamydomonas cells, so it's a green algae. This is done by the group in Martinsried. Milled the lamella, and then let's say after the reconstruction, you can see all the individual ribosomes. So it gives you very high resolution details of the cells. So here you see Golgi areas, you see budding vesicles, you see the ribosomes, and you can also see all the filaments in the cells. So that's what I wanted to share with you in the let's say in the field of cryotomography, what is happening, especially on the sample preparation side, but because the sample is critical. You need a perfect sample to get to these high resolution results. If your sample is not good, you can have the best microscope in the world. You'll never get to, let's say, to a good reconstruction and to a good structure from the molecules of interest. If you want to see and hear more and hear about all the developments that are ongoing, you can go to thermofisher.com/cryo-EM, where you can find, let's say, all the latest information for both single particle and cryotomography. And with that, I thank you for your attention. I'm happy to take any questions.